Well, thank you, David, for that extremely generous introduction. And indeed, the title of tonight's talk is Pathogen Assassin, and it is the Leeuwenhoek Lecture. And of course, as David just alluded to, Leeuwenhoek is famous for generating one of the first microscopes. Actually, it was really a super magnifying glass, in fact, as simple as that, that enabled scientists and science in general to be able to observe bacteria and protozoa and so on. And indeed, a picture of his microscope is here. And you can see how simple it is. And it's that little tiny dot there, which is a tiny, tiny, very powerful magnifying glass that enabled him to see microbes for the first time. Of course, when we talk about microscopes now, we think in terms of a typical modern compound microscope. But actually, in truth, most microscopes these days are in boxes like this. They're automated, where you can take thousands of images in an automated way, and computers then extract the data and information from the images. So I'm going to give you a journey in microbiology, my own journey in microbiology. And so I'll start with a very general introduction. First of all, microbes get a very bad press because the majority of microbes are absolutely harmless. More than that, they're essential for the life of this planet, involved in the carbon and nitrogen cycles and so on of the planet. So we should be very grateful to microbes. But unfortunately, some of them also cause infectious diseases. So infectious diseases are those that are spread either directly, somebody coughs in your face, or indirectly, maybe some rather poor hygiene, from person to person or from animal to person by pathogens. But they also come in a variety of sizes and forms. So you have viruses, of course, we have bacteria, we have fungi, protozoa, and also worms. And actually, the previous recipient of this lecture, Joanna Webster, gave a presentation, her Leeuwenhoek presentation a couple of years ago on schistosomiasis. So it's great that parasitology is getting featured twice in a row. So, as most people will be aware, the infectious disease disproportionately affects those in the poorest populations of the world. And the more orange or red the country in this map, the higher the proportion of infectious disease per unit of population across the world. You can see that sub-Saharan Africa does particularly badly in this respect, and we'll come back to that point later. So what I'm going to talk to you about, first of all, are diseases mostly caused by protozoan parasites. So that's this group here. And this is a trypanosome, and we'll be looking a little bit at the biology and biochemistry of that organism. So that organism, which is shown again here, African trypanosome, lives in the bloodstream, and it does not invade our cells at all. It lives entirely free swimming in the bloodstream and eventually gets into the cerebral spinal fluid as well and into the brain. But it never invades our cells. It's transmitted by the tsetse fly, and it causes human African trypanosomiasis and animal trypanosomiasis, which has a big economic burden. Terrible disease. Its related brothers and sisters include the Leishmania, which causes a variety of diseases collectively called the Leishmaniasis. And that's transmitted by tiny, tiny little biting sandflies. And those parasites specifically invade our white blood cells, the monocytes and macrophages that normally should be there to get rid of infections. And that parasite chooses to live inside of them. And then the South American cousin of the African trypanosome, Trypanosoma cruzi, again is an invader. It invades almost every cell type in the human body with a particular predilection for smooth muscle. And it's transmitted by these horrible biting reduviid bugs and causes Chagas disease. So Chagas disease is named after the eminent Brazilian clinician and scientist Carlos Chagas, and it's not a lifestyle choice, as some people think from the name. So I'm first of all going to give you a few minutes of some discovery science. And I've had the privilege of working with a very large number of very talented people, and they're shown on, some of them are shown here on this slide. And I can't possibly go over all of the things that we've collectively studied over many years. So I'm going to just focus on a couple of particular areas. But it all relates to this thing, glycosylation in trypanosomes. And I can see people drifting off already after having said that. So first of all, what is glycosylation? Keep it very, it's very simple. In this little diagram here, the purple bit in the middle, that's protein. And the blue bits streaming off of it here, and actually this green bit as well, those are carbohydrate chains. And glycosylation is the process of adding carbohydrate to protein. And we have some real experts in glycosylation here in the audience tonight, Tenfasi and Andel, I noticed coming in, so I'm very grateful for their presence. So that's what glycosylation is. And then the question is, well, why would you study it in trypanosomes? Well, trypanosomes are a genetically tractable eukaryote. Let me back up a second. What's a eukaryote? Germs, bacteria, they're prokaryotes, and almost everything else is a eukaryote. So we're eukaryotes, tiny little protozoa are eukaryotes, yeast are eukaryotes, and so on. So it's a eukaryote that we can genetically manipulate. We can take genes out, we can put genes in, we can mutate them, and so on, which means you can study things. And it has features which are common to all of the eukaryotes, and it has some unique features as well. So the commonality means that it's sometimes an ideal model organism to study certain types of glycosylation events, and the differences, the unique bits, may give us opportunities for therapeutic intervention. So that's the combination of attraction of studying glycosylation in trypanosomes. So, what's wrong with this picture? There's nothing wrong with this picture. It's a beautiful picture. It's a scanning electron micrograph of blood infected with trypanosomes. So these are red blood cells, the red things, and the yellow things are white blood cells, and then the gray things are these parasitic protozoa trypanosomes. What I mean by what's wrong with this picture is, why the hell are the trypanosomes surviving? Going into the human bloodstream and being able to survive is a really difficult thing to do, because we, like all animals, have developed sophisticated immune responses, both what are called innate immune responses, things that usually kill invading organisms instantaneously. And if that fails, we have adaptive immune responses that after 2 or 3 weeks produce enough antibodies or T-cell responses to kill the parasites. And yet these guys seem to have no problem at all living in the blood. So let's ask a question. Why is that? How can they survive in that otherwise very hostile immune environment? So let's take a look at one of those parasites again. Let's imagine cutting it across its cell body like that, and then projecting, looking end on at what the cross-section of the body would look like. Here's the cell body. This is actually the flagellum. I don't know if you noticed, but these parasites have a flagellum that runs all the way down the cell body, and they actually swim off in that direction. And so that's what the cross-section looks like. And if we blow that up a little bit more, and these are beautiful electron micrographs taken by Keith Vickerman in the 1960s and 70s, when actually this kind of electron microscopy was relatively new. And if we blow that up, you see these little round things. Those are tubules that keep the shape of the parasite. And then on top of that, there's a little sort of railway track. That's what's called the plasma membrane, top and bottom. And then on top of that is this surface coat. It just looks like somebody's taken a magic marker pen and drawn around it. And the thing that's unique about that is it's the fact that it's of common unique thickness and density. And most cell surfaces don't look anything like that. So why would you have a cell surface that looks like that? How could you make a cell surface that looks like that? The answer to that was solved by my former boss and mentor, George Cross, when he described what makes up that surface coat. And it turns out it's made up of a dense monolayer of identical molecules called variant surface glycoproteins. And it's because they're made up of identical units, that's what gives you uniform thickness. Now, this coat is ideal for defending yourself against the host immune system. And we might come back to that in the questions, perhaps. But one of the problems that I was given by George at the time was, okay, we know we've got a monolayer of VSG molecules protecting the parasite surface, but what's keeping that stuff stuck on the membrane? In other words, what's this thing down at the bottom that anchors these multiple copies to the plasma membrane? So that was my task that I was given way back when. And just to recap then, here's the parasite. Imagine cutting a little tiny bit of membrane off the surface and making a cartoon of it. Here's the dense palisade of VSGs that are protecting that parasite and keeping it alive in the bloodstream. Here's a single one of those VSG molecules in a bit more molecular detail. And what we're interested in is what's this thing right at the bottom, which at the time was a completely unknown structure. So what do you do if you have an unknown structure in biology and biochemistry? Well, you smash it up. You break it up into pieces. Okay? Now, we don't actually use a hammer. We tend to use strong acids and things like that. But you have to smash it up, and then you have to shake out the bits and identify the component parts. And you see, and that's using analytical chemistry. So we've gone from biology to biochemistry to analytical chemistry here. And then you can identify the component parts using analytical chemical methods, and we find that we've got a glucosamine block here, we've got a galactose block, a mannose block, et cetera, et cetera. And when we kind of color code those, green for sugary things, galactose and mannose and so on, glyco bits, and then we've got some phosphates as well, so it's a glycophospho thingy, and we've got some lipids in the form of these myristic acids, so it's a glycophospholipid. Great, that's a big step forward. Downside is there are literally billions of permutations as to how you could connect those molecular building blocks together. So obviously you have to reduce the degrees of freedom by finding physical associations between the component parts. So perhaps the most important step in this story was determining this substructure of the glucosamine, inositol, phosphate, glycerol, and the two fatty acids there. And because this is the Royal Society, I'm now going to break into some science, but only briefly, you'd be pleased to know. So here's that structure in chemical terms, glucosamine in red, and then this is inositol, phosphate, glycerol, and the two fatty acids. And so let's imagine that's the black box now. So, what do you do to determine that structure? Well, you treat it with various different enzymes and various different chemical treatments. So, for instance, if you treat that black box with sodium hydroxide, 2 myristic acids fall out. So, okay, that means we've got 2 ester-linked myristic acids attached to the black box. If you treat that with phospholipase A2 from snake venom, it releases just one of those 2 fatty acids, and that tells you that it must be part of this structure with a phosphate and a glycerol. And if you treat it with PIPLC enzyme, this whole diacylglycerol falls off, and that tells you the head group has to be inositol. And if you treat that with nitrous acid, which is a specific reagent for reacting with glucosamine, it releases the intact phosphatyl inositol, and therefore that has to be the structure. Okay? So in 4 easy moves, we've determined that substructure, which is a key step in determining the whole structure. What I'm not telling you about are the 20-odd treatments that told me absolutely nothing. So you can make things look very clever and elegant post facto. So we found those associations, and now we're down to the millions of possible permutations, which is better than billions, so that's a step forward. And what you have to do next is you have to get one of these. This is Steve Homans. He's a nuclear magnetic resonance spectroscopist, and if that sounds clever, it's because it is. And together with Steve, we were able to complete the whole of the structure, and indeed, at the same time, the three-dimensional structure as well, which is a nice addition. So, briefly, what we did, we had a parasitology problem, what holds the coat on the surface, and what we ended up doing was discovering a new biological chemical structure, which we call GPI, glycosyl phosphatyl inositol. You'll be pleased to know I'm going to call it GPI from now on. And this was a sort of a pivot point because it was a gateway into also figuring out the chemical structures of lots of other surface molecules of other parasites, because the methodologies we developed were transferable to that. And as you'll see in a moment, it was also a gateway to defining these GPIs as a general biological property of eukaryotic organisms, and David very kindly mentioned that in his introduction. So, to cut a long story short, by doing those kind of chemical and physicochemical analyses on the major molecules of the African trypanosome, the Leishmania parasite, and the South American Trypanosoma cruzi trypanosome, we were able to figure out what the major surface molecules look like, and also figure out their copy number, which enables us to make little cartoons of what a little section of their cell surface must look like. And you see they look quite different. And that's because these parasites have very different life cycles and lifestyles. As I said, Leishmania only infects macrophages. T. cruzi can infect multiple cells. T. brucei never invades cells. And these cell surfaces are ideally set up for that necessary part of their life cycle and lifestyle. huge amount of work there. I'm not going to go through all of these names, but these people did all the work in terms of all the chemistry and biology necessary to determine these complex structures. The two at the bottom, Lucia and Jose Previato, were not part of our team, my team, at all. They are totally independent workers from Rio de Janeiro, but I really want to acknowledge their contribution to this general field because they've done really beautiful work in this area. Okay. So I show you there 3 different cell surfaces. It gets even more complicated, I'm afraid, because in fact these parasites don't just exist in one life cycle stage. They undergo transformations between their different environments. So the bloodstream stage of the parasite living in the blood has a surface that looks like this one. But when the parasite is taken up by the tsetse fly in a blood meal, it differentiates, it changes its biochemistry, and it changes its surface architecture as well. And the surface of the insect-dwelling form of the same parasite looks completely different. It has this spiky and more sparse surface coat. And fascinating structures they are too. Why is that? Well, the parasite in the insect vector doesn't have to deal with a set of immune responses, but it does have to deal with digestive enzymes of the enzyme gut, of the insect gut. And this is a very protective surface for that kind of situation. So you can see how the cell surface molecular architecture maps with what's going on in the life cycle of the parasite. And actually comparing between parasites can be quite interesting as well. So I've just shown you the surface of the insect-dwelling form of the African trypanosome. Well, here's the surface of the insect-dwelling form of the Leishmania parasite. And at first sight you say, well, they look quite different, until you think, well, they're both kind of spiky surfaces, aren't they? And then you look at the length of the molecules coming out of the membrane, and they're about the same length. It's just that this one is made of phosphate and carbohydrate, and this one is made out of negatively charged polypeptide. So what's happened here is a bit of convergent evolution. Okay, these parasites diverged about 120 million years ago, and then started colonizing their different insect vectors subsequently. But they've come up with pretty much the same physicochemical solution for living inside an insect gut, but by using completely different component parts. So, I've told you a little bit about the discovery of the anchors and the biology and the determination of parasite surfaces and how that has provided a framework for other colleagues working in all different aspects of parasitology to kind of map onto that framework things that they've been studying, like how these parasites are interacting with other cells, be they human cells or be they insect vector cells, and so on. Without that framework, really, it's very difficult to formulate hypotheses and perform research to do with surface-surface interactions. So it's been pretty useful, I think, altogether. The other thing I'm going to mention briefly then is this gateway to defining GPI as a general biological property. And the first evidence that that was going to be the case came from so this is the first anchor structure that Steve and I solved. The second one we solved in collaboration with the late and great Alan Williams from the University of Oxford, fantastic molecular immunologist. And the GPI structure in that organism, in the rat, was slightly different in terms of the sort of appendages, if you like. But see the pink box? That's a completely conserved structure from the end of the protein to the beginning of the lipid. It doesn't matter whether you're a mammal or a mere protozoan, you're using the same structure. And since then, many, many different structures have been solved from right across all the clades of eukaryotic evolution, and that's always completely conserved. So it's a general phenomenon in eukaryotes, which makes it like a textbook fact. And this is how proteins in eukaryotes can be attached to membrane bilayers. There's really these 5 different ways. I know it says 6, but please ignore that. There are 5 different ways, and one of those is via GPI anchors. So before this discovery and this then sort of transformation out into understanding that it's a common phenomenon in all eukaryotes, we were blind to the fact there's a whole families of proteins anchored to membranes in this way, and it has altered quite a lot of the way biochemistry and biology has been done in numerous systems. One of the funny properties of GPI anchors in mammalian cells, in our cells, is that they sequester themselves in these things called lipid rafts. And Chitu Mayor is in the audience, and if I get any questions on those, they're going to you. So these are sort of specialized little subdomains that float around in the surface of cells. Actually, they're constantly dissolving and reassembling. But the best markers for them are actually GPI-anchored proteins. And so it's played a big role in the discovery of lipid rafts and discovering the functions of lipid rafts, which include some very important things in biology like signal transduction in the immune system and so on. But all well and good, but back to parasites. So all this is very interesting for biology, that you've been able to solve all of these structures and found that it's a common pathway in all eukaryotes, including human beings. But does that make it a problem for potentially being a target for making a medicine? Because if we're using the same structures and pathways as the pathogen, doesn't that mean that it'll be impossible to make a medicine targeting that process? Well, the first evidence that might not be the case, that it might be possible to make a medicine, came from work from Lucia Guther, who's my lifetime partner in science and in life, my wife. And I'm not going to go through the details of this. These are biosynthetic pathways in the pathogen, Trypanosomatid brucei, and the host, the human beings. And just take into account that there's a blue reaction and there's a pink reaction. And in the parasite, the pink reaction always occurs before the blue reaction, and in the human, the blue before the pink. And that tells you that there are exploitable differences between the human and pathogen pathways, or potentially that there are. Now, one of the problems of discovering something new is there aren't any reagents for it, and you have to make them yourself. So I had a wonderful set of years with Professor John Brimacombe, who's a fantastic synthetic carbohydrate chemist, and Sylvain Cotas, and also Art Crossman, chemically synthesizing these early biosynthetic intermediates and making lots of derivatives thereof, including this one, which turns out to be a cell-permeable, selectively trypanocidal GPI pathway inhibitor. So this was really a chemical validation that you can make chemical matter that affects GPI biosynthesis only in the trypanosome and kills the trypanosome and doesn't affect human cells. And Terry Smith was involved in all of the biochemical and biological testing in those studies. Okay, so there's hope that pathway could be targeted for drug discovery. And that brings me on to the issue of translational science. So what do I mean by translational science? That's the process of taking fundamental discovery science and moving it towards societal benefit, actually, in this case, new medicines. And so it goes back really to 2002, and at this point I'd like to introduce my partner in crime, Alan Fairlamb, who's in the audience. And back in 2002, I met Alan walking down the corridor looking very glum. I said, "What's the matter?" He said, "I can't get a certain pharmaceutical company that will remain nameless to tell me what the outcome of a high-throughput screen that I've just done with them on a trypanosome enzyme. They did it, but now they won't tell me what the answers were." And I said, "Yeah, it's really a problem, isn't it, getting any traction?" So just to say something very clearly, back in the early noughties, the pharma industry was really not very interested in neglected tropical diseases. It has no shareholder value whatsoever, and so that was a perfectly good reason. But I must say, things have changed enormously, and now they're very, very much part of the solution, particularly companies like GSK and Merck and so on. Okay, so we realized at that time that, if we couldn't take our opportunities to the pharmaceutical industry to develop medicines, which is kind of the classical way of making medicines, maybe we'd have to bring pharmaceutical company expertise and capability into the university. And we did that over the next 4 years and founded in 2006 what we call the Drug Discovery Unit, which is celebrating its 20th anniversary last week, actually. And that's what we did. It sounds like a simple thing, but at the time it was quite radical. People weren't really doing drug discovery in an academic environment, and there were a lot of naysayers and dissenters who said that we shouldn't be doing it. But anyway, we set off to do it anyway. And whilst it's been a great experience for Alan and myself, I'd like to point out that we were the instigators, we were not the drivers of the success of the drug discovery unit. For the next several slides, I'm going to bask in the reflected glory of the people who actually built the drug discovery unit, performed the work, and created the new medicines. So from 2006 to 2022, it was led by Paul Wyatt, who I'm pleased to say is in the audience, and then taken over from then by Ian Gilbert. Ian Gilbert's also been part of the drug discovery unit from the beginning. Also, big call out for Julie Frierson, Kevin Reed, and David Gray, who were also some of the early instigators of the drug discovery unit. These are all top drug discovery professionals from industry. Okay, so how do you get to a medicine? Well, first of all, you start with an idea from discovery science. Big message for everybody, especially funders: never give up on discovery science. If you don't have any discoveries, you haven't got anything to translate. That's a mistake. So it's very important to support discovery science. So basically, how do the bugs tick? How can you find an Achilles heel to maybe attack them? But then that alone is not enough. You need to have translational science, that's where the drug discovery unit comes in, to convert those ideas into candidate medicines. And they're only candidate medicines because then somebody has to do preclinical and clinical testing to see whether they're safe, whether they're efficacious, and whether they're better or worse than any existing therapies. Now, in our case, the downstream partners that help us with this activity are product development partnerships, PDPs, like the Drugs for Neglected Diseases Initiative and the Medicines for Malaria Venture and the TB Alliance, but also huge help from our pharmaceutical partners like Merck and GlaxoSmithKline. There's another hurdle of product registration, but we won't go into that. We'll go straight back to the drug discovery unit translational bit. So if you want to build a drug discovery capability, you need a lot of bits, a lot of component parts. It's very multidisciplinary. This is what I've really enjoyed about my experience of being exposed to this kind of activity, is just how multi and interdisciplinary drug discovery is. So you need to have methods of selecting and assessing targets and assays that you're going to put in the front end. You need lots of robotics and you need lots of compounds. We have, I think, 350,000 compounds in our compound library now. You need to be able to build sophisticated assays for both molecular targets, targeting a specific enzyme, a specific thing, or what we call phenotypic screening, just asking the question, which compounds kill the bugs? So cellular assays. You need a huge amount of data management. You need lots of biophysical methods like crystallography and cryo-electron microscopy. You need lots of computational chemistry. As I say, you need compound libraries. You need in vitro, that's in test tube, and in vivo, in animal models of the disease. And you need this, DMPK, drug metabolism and pharmacokinetics, understanding of how molecules actually behave inside the body. Because if you can make what looks like the perfect drug, but if it's metabolized in 10 minutes of taking it, it's useless. So DMPK is an incredibly important component. Actually, the first thing that Paul Wyatt did when he came to set up the drug discovery unit was he said, Mike and Alan, we need to insource the DMPK. And that's what he did with Kev Reed, and that was the best decision ever made, I think. Okay, so what happened next? Well, that was in 2006. By 2010, Paul and Julie Frierson and this bunch of other fantastic scientists had produced a thing called an N-myristoyl transferase inhibitor. So this was a molecular target which we believed, if we were able to inhibit it, should kill the African trypanosome. Lovely collaboration with Debbie Smith from York University and the Structural Genomics Consortium from Toronto. And indeed, they made a beautiful molecule that does exactly that. In fact, here's a normal healthy trypanosome. You add this drug and see it blows up into this balloon-like thing. Here's the balloon shape here. And they just burst open and they die in a most spectacular fashion. Great, fantastic. So what could possibly go wrong? This failed, unfortunately. Okay, not because the science wasn't good, not because the target wasn't good, but because we could get to a molecule that could be a clinical candidate for the stage 1 of the disease when the parasite is in the bloodstream, but not for the stage 2 of the disease when the parasites are in the brain. So it failed for a very specific reason. It's not all bad news because this was actually repositioned into leukemia. It turns out the same enzyme is essential for certain types of blood leukemias, and it's been fast-tracked by the Food and Drug Administration in the States into phase 2 clinical trials for refractory acute myeloid leukemia. So something very, very good is likely to come out of this. As I say, keep on saying to everybody, there's no substitute for activity. Okay, so disappointment there, but success to be described in a moment. The other thing, though, before I shift from African trypanosomiasis to Leishmaniasis, is that if you want to eliminate a disease, and that's exactly what the WHO wants to do by 2030 for human African trypanosomiasis, sleeping sickness, not only need you safe and efficacious therapies, you also need appropriate diagnostics. You need to know who to give the medicines to, particularly when the early symptoms of sleeping sickness are rather like malaria. Malaria is very, very common. You need to be able to decide who should get the sleeping sickness medicines and who should get the malaria medicines. And so this is where the work of Lauren Sullivan comes in. She's also, happy to say, in the audience. And here she is giving Lord David Willits a prize, or perhaps it's the other way around. I can never remember. But here we've got Conservative minister giving a prize to now Labour MP for Gravesham. So fortunately, nobody died, so that was good. Yeah, so I'm very proud of the fact that Lauren is now a Member of Parliament. So she used a very sophisticated set of methodologies, quantitative proteomics, to find what might be good diagnostic antigens for trypanosomiasis. And then she built a prototype with colleagues in collaboration with Mark Carrington from Cambridge and Steve Wall from BBI Solutions, these prototype lateral flow tests. Now, I used to have to spend ages explaining to everybody what a lateral flow test was. But I can't skip that now. The only difference is instead of putting your grubby thing from your nose onto the pad, it's just a drop of finger blood instead. But obviously, if you're infected, you get a couple of bands come up on the lateral flow test, and if you're not, you don't see anything. So it's a very, very simple, very low cost, rapid diagnostic test. So in summary, here we have basic science, molecular parasitology, advanced technology in the form of proteomics, quite a lot of statistics in this too, industrial collaboration with BBI, all mixed together, and then we give the intellectual property free of charge to a downstream product development partnership organization, in this case FIND, Foundation for Innovative New Diagnostics, and they get it commercialized. It's now made by Abbott and provides impact for patients. So I think it's a really nice example of interdisciplinary science leading directly to patient impact. Okay. So I left you on a bit of a downer with the sleeping sickness story drug, but better news is that the Wellcome Trust recognized that although it didn't get to the clinic, it was a superb piece of work, and that, guess what? These people in academia can make drugs. And so they gave Paul and his team more money to make more drugs. And I'd like to recognize Manu Duraika and Tim Mars here, because this has all been done in collaboration with GlaxoSmithKline, the GSK labs at Tres Cantos in Spain. And Tim leads the team there, and Manu leads the Kinetoplast team in Dundee. And these guys and the whole of the team work as an integrated unit. It's a fantastic example of collaboration. And over the years, they've produced two, not one, but two potential drugs for Leishmaniasis. This one that inhibits a thing called a protein kinase, and this thing that inhibits a thing called a proteasome. And they are both completing phase 1, that safety, clinical trials in humans as we speak. So it's a tremendous output. Now, how do we know that this one works on the proteasome and this works on a kinase? Is an interesting question, because we didn't start with those targets. We started with the bug. We found compounds that killed the bug, and we optimized them against the bug, and then we said, ooh, wonder what they actually interact with. And this is where Susan Wiley's group really comes to the fore. Professor Susan Wiley, former postdoc of Allen's, actually, and she set up a mode of action group that's really, frankly, second to none. And they can determine the mode of action of compounds really well by using very sophisticated mass spectrometry and genome-wide technologies. In fact, in the last 10 years, she's solved the modes of action of more than 70 compound series, including 5 clinical candidates, including the last 2 that I just showed you, with about 85% success rate. And that's really something. And here's that team when it was at its height at around about 2016, both the Tres Cantos Spanish team together with the Dundee team we came together for a scientific conference and celebration. There's Susan, standing in the middle. There's Manu hiding at the back, but there's another picture of him. There's Tim Miles right there. And also like to call out Pepe Fiandor, who was the original boss of the Spanish site, who retired a few years ago. Absolutely essential to get this collaboration going. And it's won many actual awards and recognition for industry and academic collaboration, this particular one from the Biochemical Society. And I think that's great, and we need to see much more of that. There used to be much more of that than there is now. And I think it's a shame that that has reduced. Okay, how am I doing for time? All right. So I'm going to tell you a little bit now about what the team have done for malaria. Now, malaria is a really serious disease, as I'm sure you all appreciate. Over 250 million cases a year, 600,000 deaths a year, mostly children under 5. Also, high fatality rate in pregnant women, mostly in sub-Saharan Africa. Okay, and the even worse news is that the main frontline drug, artemisinin, which was discovered in China from a herbal remedy, is a fantastic drug, but resistance to it is now emerging in the malaria parasites. It's very prevalent in Southeast Asia and appearing more and more rapidly in Africa. So that's a problem. So we need new drugs to replace the old drugs that are failing. Now, the majority of anti-malarial drug discovery and development is coordinated by this PDP in Geneva called the Medicines for Malaria Venture. And just to avoid any conflict of interest, I used to be on their board of directors, so I'm bound to say nice things about them, but they really are a fantastic organization. Okay, so I'm going to use this to make this point that making a medicine isn't easy, and the science and technology that goes into it is very considerable. You start off with a very early opportunity, something which we call a hit, which does the job, but it probably isn't do it very well, and it probably has some other liabilities in terms of a medicine. So over the optimization process, you need to increase its potency, make it better and better against killing the parasite in this case. You absolutely need to increase the drug solubility, because guess what? We're made of water. So if you want to get a medicine into a person, you better make sure that it's got good water solubility. And of course, you need to reduce its toxicity. Chemicals have a horrible habit of giving you side effects. You need to engineer those out. And you need to adjust the drug metabolism, how quickly it's destroyed by the liver and excreted from the body. Generally speaking, you want to make the residence time of the drug as long as possible. So you want to optimize all 4 of these things at the same time. And it's like one of those puzzles where you're trying to get 4 ball bearings into the center. You've got 3 in, and the 4th one pops in, and then 2 other ones drop out. It's just like that. And I think Edvard Munch must have been exposed to this problem when he painted the screen, because that's pretty much how it is. Okay, so here's Beatrice Barragana, who has led the malaria team in the drug discovery unit for many years. She's a fantastic person and a fantastic medicinal chemist. And she was the person responsible for taking this not very good, but decent hit compound and improving it step by step. So she was able to take a molecule that wasn't so good and improve it quite a bit, so that became what's called an early lead. She's basically taken that bromine off and replaced that group in this case. And then with another round of medicinal chemistry, she's been able to optimize it still further. And suddenly all of these parameters go green, and you've got a drug candidate. Ta-da! She made about 300 compounds between here and here, just to be clear. And that's actually not a particularly large number in a drug discovery campaign, but that was a fantastic piece of work. So again, big article in the scientific literature. Here's the Dundee team that made that molecule. There's Beatrice there, sitting there. And this is a multi-stage anti-malarial agent. Now, why is that important? Well, malaria goes through a very complex life cycle between the mosquito into the human being. It goes to the liver, replicates there for a while, comes out of the liver, goes into the bloodstream, and then replicates in the red blood cells, which is what gives you your fever and your frank illness, and then it differentiates into other forms that get taken up by the mosquito, et cetera, et cetera, et cetera. And this drug kills all of them. That's what you need to know, and that's a really useful property. So here's an anti-malarial agent with multi-stage activity, has a novel mode of action. It inhibits protein synthesis. No other antimalarial does that. That's a really good thing, because if you're using a drug and the parasite becomes resistant to the target of that drug, very likely every other drug that you make against that target is also going to fail. So if you find a novel target, that gives more longevity to your compound, your drug series. And indeed, in human infection volunteer studies, yes, people do volunteer to be infected with malaria. It also demonstrated that it could cure malaria with a single dose, and that's pretty unusual. And also, it was shown that a very low dose of this molecule will prevent people getting infected by malaria with a very low dose for a whole month. You take one small dose of this drug and you're protected against getting infected, chemoprevention, for a period of up to actually over a month. So that's why Medicines for Malaria Venture loved it. It's why they partnered it with Merck KGaA, the drug company, who've taken it through both phase 1 and phase 2 clinical trials, and looks like it's doing extremely well. And last but not least, Paul was also responsible for setting up a tuberculosis drug discovery campaign in the drug discovery unit way back when. And over the years, over 1 million compounds have been screened against tuberculosis, not by us, but by colleagues in the United States. But what Laura Cleghorn and her team have done is optimize those towards new medicines. And she has been able to make 2 what are called preclinical selection candidates over that period. And one of those, we're pretty sure, is about to go into human clinical trials, hopefully this year. So that's really tough stuff. It's very hard to make medicines against TB. It's an important pathogen. 10.7 million infections a year, 1.23 million deaths from tuberculosis every year. Yeah, it's a lot. Okay, so 20 years of the drug discovery unit. What has it achieved in terms of making anti-infective agents and diagnostics? What has it delivered? Well, I can very proudly say, and again, basking in the reflected glory of the colleagues who've done this work, that the DDU has created in that time 1/4 of all the new antimalarial drugs currently in clinical trials in the world, 1/4 of all the new anti-Leishmaniasis drugs currently in clinical trials in the world, 1/4 of all the new anti-tuberculosis drug candidates in the last 6 years, and 1/2 of all the current rapid diagnostic tests for African trypanosomiasis. And we think we can be very proud of that. But I'll make a political point here. What are those numbers? That's 1 drug, 2 drugs, 2 drugs, 1 diagnostic. It's not really good enough, is it, planet Earth, for diseases that are affecting so many millions of people. And basically the answer is we need more activity, more international activity in this area. Unfortunately, we're sailing geopolitically into a time where we're going to be spending much more money on things like weapons, and I fear that the impetus in this will be, if anything, diminished, but let's hope that that's not the case. Okay, I mentioned earlier that it's the global south that suffers mostly from these neglected infectious diseases. And I want to also call out the training team that the Drug Discovery Unit has. Again, all credit to Paul White. It was him that insisted that we should have a training element. And this trains scientists from disease-endemic countries. Beg your pardon. They either come to Dundee and receive bespoke training. Short courses are also run in South America and Africa. The team have helped set up 2 drug discovery hubs, one in Africa and one in Brazil, and they have lots of fantastic online material and so on. Because ultimately, all or most of this activity should be shifting to the global south, which are the disease-endemic countries. All the clinical trials are run there superbly well because that's where the disease is, but they should be running their own R&D and their own manufacture as well. So that's going to be part of the transition that we'll see in the world of a movement of this kind of work from global north to global south. Okay, how am I doing? Okay, need to go really fast here. So it's a journey in microbiology, and the journey has taken me into some really odd places. And so I'm now going to move away from sort of science and drug discovery, just to make a couple of comments about some of the other things that fine messes it's gotten me into. And one is building scientific infrastructure. And I think I know more about architectural plans and things than I ever wanted to. So you do hit problems when you're developing these kind of activities. The drug discovery unit started quite small, grew to about 40-odd people, and then Paul and Julie Ferguson at the time came to me and said, Mike, we're running out of space. That's okay, I'll go and build another building then, foolishly. And so we did. But, you know, it's a lot of work, and this is the list of donors here, huge amount. I want to call out Morag Martin, my colleague, who has helped me with all of this fundraising and so on. But we managed to do it, and we built new drug discovery labs as well as proteomics labs and computational biology labs and so on. And it was opened, and I thought I should show this, by the current president of the Royal Society, Sir Paul Nurse himself, with Brian Cox, also known as Logan Roy from Succession, as the master of ceremonies. And it wasn't actually as sweary as you might think. Okay, great stuff. What about the other thing about scientific infrastructure is you also have to think about economic development. I live in a beautiful city, really fantastic city, Dundee, but it is economically challenged. And we have this opportunity, I think, to do something about that. So the drug discovery unit, as well as doing all of that lovely work in infectious diseases, also works on other more commercially sensitive things, and it has lots of industrial partnerships. It's also enabled a whole slew of spin-out companies. In Canada, assets moved to the United States, in Germany, in Canada, and one still local. But we've been losing our spin-outs to elsewhere. And we've also been spinning out other wonderful companies like Exscientia, was actually did one of the largest IPOs, intellectual property offerings, of any biotech company back in 2021. And my colleague Alessio Ciulli, brilliant guy, founded Amphista Therapeutics, also a super successful company. But that went to Oxford and is now to America, and that one went to Cambridge. And I was thinking, wow, would it be nice if we could keep those jobs in Dundee? So we, over several years, again, Morag again, managed to raise some money from these multiple funders again to build what we call the Innovation Hub, which is actually purpose-built space for biotech companies to occupy and grow with nice office and laboratory space, just opened last year. And the idea here is to provide quality jobs for our school leavers and our graduates in the city, and therefore boost the economic well-being of what is quite an impoverished post-industrial region, basically to deliver for our key stakeholder, the taxpayer. And last, the other thing that my journey in microbiology has enabled me to do is to get involved in scientific governance. Whoops, wrong way. And I've had the absolute privilege of working on the board of Wellcome and its wholly owned subsidiary Wellcome Leap, and interacting with some truly amazing people. I'm delighted to see that Julia Gillard is in the audience today, but the chief executives like Mark Walport and Jeremy Farah, for example. Really amazing people, and also at MMV, and now with UK Biobank, Medicines Discovery Catapult, and a little biotech called Phaser. And I've learned so much from interacting with these super talented and driven people, and Rory Collins from Biobank is kindly in the audience today. So it's been an interesting journey, and there are many, many people to thank along the way. My sort of key mentors and employers at one time along the way were George Cross and Raymond Dweck in Oxford, Phil Cohen, who recruited me to Dundee in 1988, something I've never regretted. It's been a fantastic place for me to learn my craft and to grow. And Sir Alan Langlands, who was former principal of the University of Dundee, we really encouraged everybody to think large about what they might do. And my great friend and confidant, Doreen Cantrell, who I know has come down for the talk. And then there's the scientific collaborators. Alan Fairlam, who I introduced earlier, co-conspirator in establishing the Drug Discovery Unit. Lucy Guth, who basically ran my lab for 25 years. And then the three individuals who set up and founded the Drug Discovery Unit, which I've talked about so much tonight, and Morag Martin, who's helped build the infrastructure to house all of that activity. And with that, I'd like to thank you all very much for listening.
Mike, thank you so much for a truly awe-inspiring lecture, and I hope you'll agree to take a few questions now. And I hope you'll agree to take one from me to start with.
God.
You referred to the question of longevity of these drugs and the art of medicine in resistance breaking. And I wondered, what is the thinking? I mean, you described how, I mean, it's a huge effort to get just a single drug.
Yeah.
And it's almost an inevitability that at some point, a drug that gets widely used will be overcome. So what's the thinking about avoiding this Red Queen effect?
Yeah, so the strategy at the moment is you're not allowed to register a single entity drug for malaria anymore. Hasn't been possible to do that for some time. So in other words, it always has to be a minimum of a dual therapy. So when you have two drugs, then the probability of getting resistance to either one is reduced enormously, because basically even if in one infection you become resistant to drug A, drug B is still going to take care of business and take those parasites out. And indeed, MMV are moving towards triple therapy is probably going to be the standard of care as well. So the best way to protect a drug is to put another drug with it.
Okay, thanks very much.
You're welcome.
So you choose the questioners. So there's one in the middle there.
Hello? Yep.
So Mike, that was a lovely talk. I want to pick up on two things I think are related in your talk. One, you said there's been a reduction in academic industry collaboration throughout your career.
Yeah.
And the other one is that very sad slide at the end when almost all your spinouts went west.
Yes.
What are we getting wrong as a country?
Oh, right. Okay, well, I think... let me take the latter part first. What are we doing wrong as a country? We have to, us up north, we have to deal with both conscious and unconscious bias. All right? So if we're looking to get inward investment from venture capital into our spin-out companies and so on, frankly, we have to jump through more hoops than you do if you live in the Golden Triangle. Now, we can moan about that, or we can just jump through those hoops. So that's fixable. We can just do go those extra miles. But what would greatly help that would be, here's the discussion. You've got a great idea. Maybe you've got 4 or 5 potential new companies, and you pitch them towards a venture capital house. And they go, oh, well, we really like the first one and the third one. Oh, those are both really interesting. Come back when you've done A, B, and C, and X, Y, and Z. And everybody goes, well, who the hell's going to pay for that? Right? And the private sector expect the public sector to pay for it and vice versa, and it's just a Mexican standoff. So actually, if UK PLC wants to do something, one thing, that would really liberate a lot of IP that's stuck in universities, it would be to create a proof of concept fund. I call it Innovation to Investable Asset Fund. It doesn't even have to be very big. For £250,000, you can take that thing which is of interest but not quite there yet with the venture capitalists and make it investable. And then you're off to the races. And we need that particularly outside of the Golden Triangle, but actually the whole of the UK could do with it. So that's one thing. And I'm so sorry, what was the first part of your question? The collaborations. Collaborations with industry. Yeah, it used to be, years ago, the industry were very keen to give compounds out that they were developing so that scientists could see what they were doing in their systems and so on. I'm looking at Keith Gull, who did a lot of that with antifungal agents and so on. And it just stopped. And companies became very cagey about giving out any of their compound series to academic collaborators. And I'm not entirely sure anybody really understands why, but it just happened. Yes.
Thank you. So I grew up in the United States in Missouri and work at the University of Missouri in a capacity of biochemistry. And the infectious disease of Chagas disease, the Trypanosoma cruzi has been found in Missouri for the last few decades in testing of the intermediary host insect, and yet both the State Department of Health and the CDC refuse to recognize any endemic transmission in the United States, and there is no approved treatment for adults that contract Chagas disease. Why do you think that there might be some resistance towards recognizing this, despite scientists and physicians in the United States seeing this rise in these previously tropical infectious diseases that are moving to more temperate areas?
Yeah. Well, I'm not here to be an apologist for the state systems, but clearly there is lots of Chagas disease in the southern parts of North America, Texas as well in particular, and clearly you have some of the Reduviid bug vectors as well, and you have Chagas disease, some of which has come from migration and so on, some may be endogenous. So it's definitely there. I think part of the problem is there isn't a safe and easy treatment. So the two frontline drugs, benznidazole and nifurtimox for Chagas disease, are bloody awful. And unless and until folks like my colleagues make much safer and efficacious drugs, I think everybody's reticent to really do anything about it and therefore to recognize it. It's a bit like toxoplasmosis in the rest of the world. It's just generally not treated because we haven't got anything that's particularly good at treating it, and the drug may be worse than the disease in that particular case. But Chagas disease is a very serious condition, killing about maybe 10% of the people who become infected many, many years after infection, as you know. Pleased to say that actually, I didn't discuss it tonight because the compounds are not over the line yet into clinical trials, but the Drug Discovery Unit, Manu Duraika's team, have two compounds which are looking really promising as anti-Chagasic agents, as indeed does the drug company Novartis as well. So it may be that in the next 5 to 10 years, there's a much better treatment for Chagas than there has been.
Thank you. And I was also wondering, do you have advice for people who are just looking to start careers in pharmaceutical development?
It's a great career, and there are wonderful pharmaceutical companies to work for, but there's also the opportunity these days to do that drug discovery activity in academia as well. Not a huge number of academic drug discovery units, but there are several now. And so you have the option of doing it in academia as well. But it's a fantastic occupation. It's all that interdisciplinary science and so on. But you do have to be prepared for the fact that the project that you've been working on will suddenly just be killed because it's not going fast enough or not going well enough. And then you'll be put on something else. So as long as you're robust enough to survive that, go do it. Liz.
Thanks. Wonderful talk, Mike. I'm curious if you could tell us about the path from a failed drug, your trypanosomiasis failure, how you get from there to curing leukemia. And is that an academic taking that path, or do you have to get pharma interested? How do you make those connections?
Yeah, it was academic to academic, actually. So the NMT inhibitors were published, and a guy called Luc Berthiaume, based in Canada, who's a great scientist, and he's been working on protein N-meristylation for years and years and years, and had this hypothesis about that being a great target for a particular subgroup of leukemias. And when he saw that paper, he said, I've got to have some of those compounds. And so he got in touch with David Gray and Paul Wyatt in the Drug Discovery Unit. Science was done, and a very nice Nature Communications paper published showing that they were indeed in vitro models efficacious, and they went straight into the clinic. Because the pharmacological profile of these molecules was so good, low toxicity, highly soluble, good pharmacokinetics, and so on. So they were able to be retest straight into another disease.
And so then, is there a way to, how many of those kinds of molecules that fail in one thing are going to waste because they're not being tested in other contexts? And is there an opportunity to make libraries of those kinds of compounds that once you have a screen in hand, anybody could test? Yeah,
so yes and no. And it's a bit of a complicated answer because, so this is called, very popularly called repurposing, the idea that you've got these molecules which are very good quality molecules and they were made for X, but they might do something for Y. And they're often very good test compounds to see whether that mechanism is efficacious in another disease. The truth is, though, they were designed exquisitely so that the pharmacokinetics and pharmacodynamics of those molecules were for condition X, and that often doesn't map on to condition Y. But they can be very, very useful tools, but often you have to go back and re-engineer a new chemotype in disease Y. So the number of successful examples of drug repurposing are far fewer than the number of reviews written about drug repurposing. I don't know whether Tony Wood, CSO of GSK, would agree with that statement or not, but yeah, okay, great.
Maybe one more question, yeah.
Thank you very much for your talk. I was just wondering, is there any appetite for biologic drugs, things like antibodies, enzymes, and oligonucleotides?
Absolutely. Any drug modality that works is wanted. I must say, we tend to focus on small molecules because the cost of goods for treating these diseases in very poor endemic countries maps on rather well to the small molecule world. But indeed, the Wellcome Trust and others, LifeArc as well, are looking very hard at bringing the cost of production of monoclonal antibodies down to a level where they can be considered for infectious disease therapeutics. My opinion, if you can kill a bug with a small molecule, that's what you want to do. But there will certainly be time and positions for biologics in anti-infection therapies. And of course, vaccines are the great example of how good biologics can be for preventing infectious diseases. So yes, but I think that sort of nexus point of the price coming down, of production coming down sufficiently, I think it's just about being reached now. So I think we'll see more biologics used in anti-infective treatment.
Maybe one more, just at the far back.
Okay, far back.
Great talk, thank you. In the advent of climate change, are we expecting a change in the sort of geographical distribution of certain tropical diseases?
Absolutely, and it's happening as we speak. Yeah. And if I may say, to the credit of my former colleagues at the Wellcome Trust, they were the first agency to create a health challenge area called Climate and Health, in recognition not only of the fact that we need to mitigate against heat so that we don't die of stroke and everything else, but also to study how climate change is affecting vector range and therefore disease range for vector-borne diseases, for example. So yes, expect lots of changes. Another example, Candida auris infections. That's a nasty fungal infection. Suddenly it started infecting people, it never used to. Maybe it's been adapting to higher temperature just by climate change, and that's helping it jump from the environment and into people. It's sort of a speculative argument. But yeah, I think expect to see lots of surprises as climate change takes hold, unfortunately.